Okay, today's topic is specific defenses and immunity. It's also called adaptive immunity or adaptive defenses. So when I think of the immune system in general, I think of kind of this concept map in my head, and I encourage you guys when you took notes from the book to create a concept map, and I put kind of a starter concept map on Canvas for you to use or not use, but I see it as like pathogens up here are things that are going to infect us. If it can get into our body, meaning bypass any physical barrier, those mechanical barriers like skin, then our second line of defense would be any kind of chemical barrier. So a chemical barrier would be like a really strong pH in our stomach, or lysozyme antibacterial enzyme created in our eyes, the salt function of sweating, so anything that can chemically stop or kill a pathogen. But then our third line of defense is our immune cells. So this is assuming the pathogen has penetrated into our body, and now we've got immune cells that are going to try to cure us of this infection and get rid of the pathogen. And so within the third line of defense, we're going to have a couple different types of cells. We're going to have our kind of generalized cells that are like macrophages, neutrophils, monocytes, things that just can sense a cell, pathogen cell that doesn't belong, and can like engulf it and wipe it out. But the other side of our cellular immune system is going to be our B and T cells, and our T cells or our B cells in particular are what can eventually give us immunity, but it takes the help of T cells also. So we're going to talk about all these special types of cells with our specific immune defense, and getting a full-blown immune reaction from our immune system means we have to recognize the exact like species and strain of the pathogen infecting us. So not just we're sick with something, but like what exact pathogen is infecting us. Our body recognizes that, and then it caters this perfectly built immune response for that specific pathogen. And the idea is, once you've had this full-blown immune response, including our B and T cells, we've created this huge army of specialized cells that attacks that pathogen, gets rid of it, and then we have memory B and T cells that will remain in our body potentially for our whole life, depending on how strong the immune response was to that particular sickness or pathogen, and you should not be able to get infected with it again. All right, so specific or adaptive defenses or immunity. This, again, like I said, is our third line of defense. And we are going to specifically respond to the pathogen infecting us. It's not just trying to keep out germs in general. It's like exactly what is infecting us. We're going to have our immune cells bump up against these pathogens and feel their little protein antigens, which is how our immune cells recognize these foreign pathogens, and figure out how to respond appropriately to that, that pathogen. So an immune response is based on the ability to distinguish molecules or antigens. And we talked about this with blood. So I think the easiest connection there is to think about our red blood cells, how we talked about if you have an A or a B or an AB or an O blood type. What distinguishes those blood types from each other are the antigens or proteins that are located on the surface of those red blood cells. So like a positive red blood cell has an A antigen and it has an Rh antigen, or a B negative red blood cell just has that B antigen. So it's got a specific shape antigen on the outside of it. Absolutely all cells have these like recognizable antigens, including our own cells. And so our own cells' antigens help our self cells recognize each other. And a non-self cell, then recognizable by its foreign antigen on that cell surface, is then recognized by your body's immune cells as like this other type of antigen or shape on the surface of that pathogen, or on our cells that are infected by that pathogen in a virus example. And it would then mount an immune response and try to kill off anything that has that antigen present. The only way that we can accomplish this is kind of by this inventory process that happens before birth. This is crazy cool. So before you're born, when you're in the womb, obviously you're totally protected in the placenta. You're not getting any foreign exposure. The mother's blood's being filtered, and only the good nutrients are getting into the fetus. But as soon as you're born into this world of germs that your immune system is going to have to try to fend off. So before you're born, your immune cells go through all of your body's cells and kind of bump up and feel them and learn the shapes of all of your self-cell antigens. And once we have this inventory, then anything that does not fit with what self antigens are there is considered other or foreign and must be attacked. And so it takes a while as a fetus when your immune cells are going through and like inventorying, feeling, going through, learning all of the shapes that your cells have on their surfaces. But it's really important to have this function occur, because if your immune cells mistakenly think that a self-cell is foreign because it didn't learn the shape of that self-antigen, then that would cause an autoimmune disease where your immune cells would attack your own cells, and that could be very problematic. So lymphocytes, again, lymphocytes are like B and T cells, B and T lymphocytes, B and T cells. They develop what we call complementary receptors. Again, these are proteins, but they're going to have kind of the opposite shape of an antigen on their surface that allows them to differentiate between self and non-self antigens. And the really cool, crazy thing is, as your B and T cells, B and T lymphocytes, are developing their receptors, they, like each B and T cell, needs to be a little bit different in the shape of their complementary receptor, because your immune system is trying to develop its capability to recognize every single foreign antigen shape that could be out there. And so theoretically, our immune systems have B and T lymphocytes that are like set with the complementary shape to fight off a foreign antigen that has not even evolved into existence yet. So once we know all of our antigen shapes, those B and T cells develop the complementary receptors to every other type of shape out there. And therefore, we have the immune ability to get immunity to any type of pathogen out there. So our immune system has crazy powerful potential. Okay, so non-self antigens, like I was saying, a non-self antigen would be like a antigen or a little protein, most likely on the surface of some pathogen, and it would, once it gets into our body, tries to infect us, it would combine or like physically contact the T or B cell surface receptor, which would stimulate an immune response. So here is representing like a pathogen with these little yellow bumps being a foreign antigen. And I know you're familiar with these pictures because they show COVID-19 virus constantly, so I put that one in here. The COVID-19 virus is called the SARS-CoV-2 virus, which causes COVID-19, which is the disease, right? So here you can see in between host cells. So I was talking about those microscopic droplets that people can sneeze and cough into the air that linger for 8 minutes, they're saying, in the air when people do this. Here is the virus inside of its little capsule. And on the surface of the virus are these little spike proteins. So here's a spike protein, and those are really antigens that can trigger an immune response when touched by a B or T cell. And but the receptor has to actually match up with that. So when you zoom into it, what it actually looks like, it's just a protein. And so you can see the three-dimensional structure there of that protein. So again, antigens, I say usually they're proteins. That's kind of the biggest grouping, because proteins have that special three-dimensional shape we always talked about when we talked macromolecules. But it could be a polysaccharide coming, like branching out of that membrane, or it could be a glycoprotein or glycolipids. Basically, just think they're specially shaped structures that branch out of any pathogen. And pathogens, again, we're talking, could be fungus, could be bacteria, could be virus, or could be protozoan. With protozoans, I mainly think of like malaria is a good example, other parasitic things. But those antigens are these specific foreign molecules, one of these types of molecules down here, that trigger an immune response because they are non-self. They're not going to be recognizable as a self-antigen because they're foreign. They're going to trigger your immune cells to try to attack them and get rid of them. They don't belong. Okay, so again, specific immune response, it involves T and B cells. T cells, you've probably heard of killer T cells before and helper T cells. So those are two types there. B cells are the ones that make antibodies. And we'll go into the specifics today. What they both have in common are they originate in bone marrow, just like all blood cells, whether they're white blood cells, red blood cells, platelets, all are made in the bone marrow from stem cells that eventually differentiate. When these lymphocytes, B and T lymphocytes, are released into the blood, they haven't differentiated yet, but if they linger in bone marrow, they become B cells, so B for bone marrow. If they circulate and go to the thymus, then they mature into T cells. So T cell means thymus cell, B cell, bone marrow. It's indicating where they matured, but they're all made in the bone marrow, like all red or all blood cells in general. Both T and B cells, they reside in lymphatic tissue, like within lymph vessels, primarily in lymph nodes, although they can move in between nodes, in the spleen, sometimes in the liver. The filtering types of organs have a lot of them. Here, this is a diagram you can see in your textbook, and it's showing how these generic blood cells are made in the bone marrow, in red bone marrow, and then if they're released and they go to the thymus, they'd be released as just generalized lymphocytes. So they'd be white blood cells, and then they'd specialize into lymphocytes, and then a generalized lymphocyte maturing in the thymus becomes a T cell, and if it matures in the bone marrow as a lymphocyte, it's a B cell, but primarily you'll find both of them hanging out or spending most of their time in a lymph node. You can see here, lymph nodes have all these little crevices, little pockets, and the pockets are good places for lymphocytes to kind of settle into, and then this conveyor belt of lymph will pass through, lymph being that fluid that could hold a pathogen in it, or a pathogen infected cell, and the lymph would go in the afferent side and exit out the efferent side and have to pass through this area that's highly concentrated with lymphocytes looking for anything that didn't belong. So it's a great way to easily find pathogens that might have infiltrated into our body. Okay, lymphocyte functions. We know that the only way that they're going to really do their job is to activate them, because they basically sit there dormant until they're activated. And to activate a lymphocyte, they have to encounter the perfectly shaped foreign antigen that fits their receptor. And again, it sounds like so complicated, but it really comes down to like puzzle pieces, right? We talked about it a lot with enzymes. If a substrate fits into the enzyme's active site, right, then the reaction gets catalyzed. Same idea here, where we have this T cell is kind of a generic T cell, could be a helper T cell or a killer C cell. Probably in this diagram we're talking helper because it's being activated by an APC. APC is antigen presenting cell. And those are phagocytic white blood cells, like macrophages, monocytes, neutrophils are those three main phagocytic cells. And so they have encountered something that didn't belong. It didn't know what it was in terms of virus, infected cell, cancer cell, fungus, bacteria, but it engulfed it because it was not presenting a self-antigen. And here you can see it's presenting the antigen after it engulfed the foreign cell. It presents that foreign antigen on a special structure on its surface, and you can see this helper T cell has a little receptor, and it happens to fit, the antigen fits with the receptor complementary, and that would activate a helper T cell. And once that helper T cell is activated, it's like its whole life's purpose is being fulfilled. So this helper T cell was developed before we were born with this specific shape receptor, and it's been looking for the complement, its foreign antigen that would fit its receptor for its whole life. And once it encounters it, it is activated. And that activation primarily causes mitosis. This activated T cell now knows that the body's infected with the foreign antigen that it is created to defend against. And so it will go through mitosis and make identical clones of itself with the identical receptor that can then attack this specific foreign antigen. And so whereas there was only one version of this specific T cell before, there will be millions once it's activated, because mitosis is a huge response to activation. There's some other cascading effects that we're also going to talk about, but being activated is kind of the first step in an immune response. We have to find the lymphocytes, B and T cells, that have the right shape receptor to build that specific army to fight off that infection in the body. So briefly here, because we'll come back to this idea, this concept of having, this is showing B cells, because you're going to have a whole bunch of B cells that are slightly different based on their receptor. Can you guys see this one looks kind of like a goalpost, and that receptor would fit an antigen that would be like a square, for example. This one here is kind of a Y-shaped receptor, and it would fit an antigen that's more of a triangle. This one has a Y, but it's a little bit more narrow. This one has a receptor that's like a cup, and you can see it's being activated. This foreign antigen has little circular shape to it, and it fits into that cup. So this activated version of a B cell, now we had a T cell example of being activated in the last slide. Now we're looking at a B cell activation example. This B cell, finding its complementary antigen, gets activated, and like I said, its first job is to go through mitosis. One cell becomes 2, 2 becomes 4, 4 becomes 8, doubling, and eventually, as millions of these cells can be formed, most of them will specialize into what we call plasma cells. A B cell being activated, B cells make antibodies, but they don't do that directly. They're little clonal babies that mature into plasma cells, spit out thousands of antibodies a second each, and they just inundate and flood your body with these specially shaped antibodies. And you can see here the shape of that antibody specifically is the same or very similar to that original receptor of the activated lymphocyte. And so those, again, can fit with that foreign antigen. And there's many different, several different ways that those antibodies work, but one way is by coating it, because those little receptors fit exactly with that foreign antigen. Some of the cloned babies from the mitosis, this back, would be memory cells, and memory B cells would be ready to make antibodies only on a second exposure to that pathogen. So if you got sick with something, it's passed around the family, and it's a month later and your fourth sibling has it, you already have the antibodies, and if you're then exposed again by that sibling, then your memory cells can make antibodies even quicker, and that activation process is much slower. Or sorry, much faster in the second exposure. Okay, so now to get into, we're going to go a little more specific and then eventually a little more specific. First topic being T cells. For T cells and macrophages, they rely on this communication chemical called cytokines. And actually, all of our immune cells are constantly communicating with cytokines, again, a protein chemical that they can secrete. And cytokines in general create activation, meaning like turning on and reinforcing the right B and T lymphocytes for, like, depending on whichever kind of infection we have. So if it was, that SARS-CoV-2 infection that causes COVID-19, we would need to find the right B and T lymphocytes that have the right complementary receptors on them. And they would, as they're being activated by macrophages and other APCs that generally just recognize foreign cells, these cytokines get released, and it tells T cells, for example, to get activated, and then it can tell other cells to go through mitosis and proliferate, divide, and create an army of themselves in order to create this customized immune response. So the cytokines are very important And like cell-to-cell communication. It's how your immune cells mostly like reinforce activating each other and encourage cell division to build a bigger army of cells to get a stronger immune response. So cytokines are really important. The most common examples of them, you may have heard of interleukin-1 and interleukin-2. Generally, they activate other T cells and can help to activate B cells. Interleukin-1 specifically helps activate T cells. Usually the macrophages are releasing that interleukin-1 to help activate T cells. Interleukin-2 usually then causes an activated T cell to have a strong proliferation or lots and lots of mitosis. Colony stimulating factor is another kind of cytokine that does exactly what it sounds like. It stimulates white blood cell production, like all white blood cells. So having more macrophages and more neutrophils and monocytes to engulf foreign particles when you're infected is advantageous. So just having more white blood cells in general. Having that larger colony to investigate and look for pathogen in the body is going to be helpful when your body's under attack. Moving this around. Here in this diagram, I wanted to kind of show an example of interleukins being released. Interleukin-1 and 2 are kind of general categories. This stuff gets a lot more complex when you really get into it, but here you can see an example of a fungus, Candida fungus, that's infiltrated the body, and you have a macrophage or some kind of phagocytic cell that's encountered this fungus that doesn't belong. It does not recognize its antigens as self. It recognizes them as foreign. So the first thing it does is releases some interleukin. It's going to help to signal white blood cells to come and cluster around it so that the cells can see if they're the right receptor to fight off this particular pathogen. And so interleukins will also communicate with other immune cells to recruit more help. It looks like here we found the right lymphocyte to then get it activated and create a colony of cells with that same receptor. You can see this little red structure that they all have. Helper T cells are going to interact with that antigen-bearing cell directly in a cellular immune response. And so T cells oftentimes are coming into direct contact with a pathogen or with that antigen that the APC found. And I always find myself in these lectures kind of going round and around in kind of the different pathways for activation. Ultimately, there are many different pathways to activating B and T cells. However, the way to get the strongest immune response is to have an APC type cell, a phagocytic cell, engulf a pathogen, find that foreign antigen, display it on its surface. We're going to then activate a helper T cell by contact with that APC, holding the foreign antigen out. And then once the helper T cell is activated, it can cause a strong immune response in a B cell and other killer T cells as well. But that idea of a helper T cell indicates that that kind of step in this activation process is how we help get the strongest immune response. So a helper T cell interacts with that antigen-bearing cell directly. It's a cellular immune response. If you ever have an immune cell that comes into contact directly with the foreign antigen, we say it's a cellular immune response. And then cytotoxic T cells are killer T cells, same thing. They secrete toxins, something called perforin, that actually pokes holes in foreign cells or infected cells of our own. And then those cells take in a bunch of fluid, they get really big and swollen, and then they burst, and that's how we can kill them. And so those cytotoxic T cells are kind of like the violent hand-to-hand combat type of immune cells in our body. They do directly try to kill off foreign cells. Another diagram here kind of showing this full-blown immune response, trying to activate our system. And again, reinforcing this idea that the helper T cell's job is very important for a full-blown immune response. And so this T with the subscript H always means helper T cell. it's going to get activated, it releases cytokines, which has a big effect on a whole bunch of types of other immune cells. Other helper T cells are going to release cytokines to get just a more enhanced immune response, kind of like sounding a red alert to all of the immune cells in the body. This helper T cell can then activate the right B cell, and that B cell can make antibodies. The helper T cell can encourage MO. This is going to be those macrophages, those engulfing phagocytic white blood cells. It can encourage them to go through proliferation and reproduce. We have more phagocytes in our body searching for the infection to engulf and destroy it. And then here, a cytotoxic T cell will also be able to proliferate, make more of them if it's a viral infection. Cytotoxic T cells or killer T cells specialize in killing off our own cells that are infected with a virus, as well as our own cells that have become cancerous. And so a cytotoxic T cell then it could kill off a virally infected cell. The cell would do that cell lysis thing where it bursts open and is killed so that we can stop the infection. My note down here, make sure I don't miss anything. Helper T cell is releasing cytokine because it has been activated, which causes B cells to become more activated, causes phagocytic cells to activate, more helper T cells will get involved. The activation process is this cascading effect to build a large multifaceted army tailored to fight off specific pathogen action. One interesting thing with COVID that I've been reading about is as our own cells become infected, usually virally infected cells send out these signals to our immune cells to like help SOS, I'm dying of an infection, come, like lymphocytes come check me out to see what I'm actually infected with. But that SARS-CoV-2 virus actually has special enzymes that prevent our virally infected cells from sending out that signal too early. And so that is that whole idea of like, you're sick with this and you could be contagious, but you don't have any symptoms yet because your cells are dying, but they're just in these little islands where they're suffering and dying, but they can't identify and share that information with the rest of your immune system to get the symptoms going and then the full-blown immune response. And so we have a lot of asymptomatic or pre-symptomatic spread because of that special enzyme that SARS-CoV-2 has. Okay. B cell basics. So B cells, we say, produce antibodies, and since B cells don't ever really contact the pathogen itself, this is our humoral immune response. Humors mean like the fluids of the body, and before we had modern science, like humors were like just any kind of liquid, and the antibodies end up being all throughout the liquids of our body, mainly lymph, but also they get into our blood and they end up being in interstitial tissue. And once we've got those antibodies, the pathogen really has not much any hope of infecting us anymore. It's kind of like a tsunami wave that goes through the body and cleans it. But since those antibodies end up in those fluids, it's called a humoral immune response instead of a cellular immune response. The best way to get a B cell activated is by a helper T cell. You won't get as strong of an immune response if we don't have help from the helper T cells, although usually B cells will have encountered the pathogen themselves before that activation or like supplemental activation from a helper T cell. So really the best way to get your B cells activated and get the strongest antibody response would be to have the right B cell encounter the foreign antigen, either by contacting the pathogen itself or by contacting an APC, those phagocytic cells that present the foreign antigen to it. And then we reinforce that with a helper T cell, and then the correct B cell is going to get activated and proliferate. You see here, the right B cell has this little Y-shaped receptor, and then once it's activated, these arrows to the next level show lots of mitosis, proliferation, cell division. We can get millions of these cells that mature mostly into plasma cells. Plasma cells make antibodies. You can see those little antibodies look like the original B cell receptor, and with their little active sites can grab onto a virus receptor cell, the virus antigen on one of our human self cells, kind of coat that, disable it, and then our body can get rid of it. We will also get some of those daughter cells from the original activated B cell as memory cells. They don't do anything during the first infection, and they just hang tight in case there's a second infection from that same pathogen. Okay, again, to reinforce this idea, every person has millions of varieties of T and B cells, meaning if you like looked at just one of your B cells and then randomly looked at another B cell and looked at the receptor it had, the shapes of those receptors are slightly different because they're customly made to fight one particular foreign antigen. And so there's a B and T cell for every potential shape out there of foreign antigen. And then each T and B cell receptor can only respond to that specific foreign antigen that has that complementary shape. So you have the potential to be immune, again, to all sorts of things that exist and don't yet exist out there in terms of pathogens. You saw this one before. Just wanted to reinforce that idea of all those different little receptor shapes. And these ones, of course, are highly simplified in real life. They're much more complex in terms of their shapes. And you've got so much more variety. But again, your body is prepared for all sorts of different pathogens out there, and it's just the process of getting the right B and T cell activated. Once you get sick, we got to find that correct B and T cell receptor to create the right clonal army to fight off this infection. Okay, so now we're going to get a little bit more specific. We're going to go down that pathway of like the best way to get the strongest immune response, and that involves those APC cells. Again, APC is an antigen presenting cell. And if you want to make like a little list of cells that fit into that category, macrophage is a big one, monocytes. Okay, when monocytes mature, they become macrophages, so those two are related, and also neutrophils. Those are the smaller but more common white blood cell in the body. Those all are phagocytic. Once they bump up against a pathogen, they're going to engulf it via phagocytosis. You can see this little cartoon guy kind of digests that foreign cell, maybe a bacteria, for example, but it saves that antigen. These cells are really smart. They can chop up and digest the foreign bacteria in this example, but save its antigen that's recognizable by our immune system. And then it has a special structure called an MHC, major histocompatibility complex, which we'll talk about, which allows them to present that foreign antigen on the outside of its cell. And what it's then going to do is release those cytokines, interleukin-1, to try to draw in every kind of helper T cell it can with every different receptor, and it'll hold out the foreign antigen and say, are you the right T cell to fight off this antigen? Does your receptor fit this shape? And it's going to present it to all the different helper T cells, trying to find the right one to get activated. And then have that one specifically multiply its army so that we can activate the rest of the immune system and have that catered, specialized immune army to fight this infection. So here shows that little MHC structure. So again, we're talking about on the surface of antigen presenting cells, those phagocytic cells. They have this special little MHC complex, which is shown here. It's a protein, and it uses that structure. This yellow glowing thing is the foreign antigen. You can see how the MHC is embedded within that phospholipid bilayer cell membrane of an APC cell. And as we hold out that foreign antigen, it's hoping to then show it to the correct T and B cell to get them activated. Here we can see that APC. Here's an APC with its nucleus. It's a macrophage specifically. It looks like a bacteria. It engulfs it via phagocytosis, digests it, and it saves that little foreign antigen. It's got a whole bunch of these little yellow MHC proteins that can grab onto the foreign antigen and present it outside of its cell membrane to hopefully find a little T cell. A little T cell comes along, it's got the receptor that fits that shape, and it can then be activated. An activated helper T cell, again, in this like preferred activation sequence, a helper T cell can then stimulate the right B cell with that same shaped receptor to be activated, and then that B cell makes the antibodies. The antibodies make for that full-fledged immune response. Here you can see kind of up close, zoomed in and personal, again, an APC with its MHC presenting that foreign antigen. Here is your helper T cell. It's got this receptor that seems to fit with this antigen. There's often secondary structures that help with this connection and the communication between an APC and a helper T cell. And specifically this one, the CD4, is a very common one. Something interesting about this CD4 structure, this protein is how the HIV virus hijacks and gets into helper T cells and then eventually destroys them and renders your immune system inactive, which is what full-blown AIDS would be. But the interesting thing is your immune system still functions pretty well without a CD4. So even if your helper T cells don't have this CD4 structure, and not having one means that you would be immune from the HIV virus. And so now what we're doing is using CRISPR to try to get that CD4 protein into people who are high risk for contracting HIV, which is amazing technology that is coming about right now. The other interesting thing, if you watch that documentary, that 4-part documentary that I put on Canvas, it's one of my favorite all year, just because of the evolutionary implications of this. This, CD4 variation of some people having a CD4 and some people not on their helper T cells. If you don't have it, you are also immune to the Black Death. And Black Death, when it swept through Europe, killed like a fourth of all of the people and at that time. And the, I guess, occurrence of not having a CD4 jumped up in a huge amount during that time because it made you immune from Black Death and now gives you immunity to HIV. I think that kind of genetic variation still lingers, as like 14% of all European descendant people lack the CD4. Anyways, another picture showing a macrophage or an APC engulfing a pathogen, digesting it, presenting it outside the cell, trying to activate a helper T cell. That interleukin-1 will help reinforce that activation. This is this like additional cell communication, helping that process along. After we've activated the helper T cell, interleukin-2 is released, and that stimulates cell division, trying to get more of that correct helper T cell, that correct receptor produced to help get a better B cell activation. So then a little bit on the other types of T cells. So besides helper T cells, there's cytotoxic T cells. They're also known as killer T cells, same thing. And they specialize in fighting off virus-infected cells and cancer cells. So our own cells that get either infected by a virus or our own cells where the DNA has mutated and those cells go rogue as cancer cells. We have killer T cells that are lurking throughout our body trying to identify that stuff early. They say that everybody gets cancer something like 10 times in their life, that we would never even know we had it because our cytotoxic T cells identify it early and we never would have had any kind of tumor or symptom from it. Our body's constantly looking for cells of our own that are not behaving correctly. So a cytotoxic T cell can recognize non-self antigens, like I said, on cancer cells or virus-infected cells. Once we activate a cytotoxic T cell by exposing them to their complementary foreign antigen, they divide by mitosis, make this huge army of cytotoxic T cells identical to themselves with that same receptor. And then the way that they can fight off these virus-infected cells, cancer cells, is once they bump up, here in this picture, cytotoxic T cell bumps up against one of our cells that is presenting like either viral antigen, viral proteins on its surface, has weird shapes on the outside or structures on the outside cell surface because of mutations caused by cancer, that cytotoxic T cell will then release this chemical called perforin, literally that can coat the outside. There's a reaction that happens on the cell membrane. related to that antigen, and it ends up like drilling little holes, and you can see kind of the dotted line of the cell membrane is now having all of these holes, little pores there, perforations, and then liquid and salts all flood into the cell. It swells really big and then bursts open, this dramatic kind of bursting and exploding, dying cell, and then obviously that cell can no longer harm the body. Again, another picture of that infected cell here. It's presenting these antigens based on what is infecting it. The cytotoxic T cell recognizes that, oh, that antigen, foreign antigen, fits my receptor. We go through mitosis, build a whole army of cells that are identical to that with the right receptor. And then when those cytotoxic T cells or killer T cells encounter cells infected, presenting the foreign antigen, they'll release the perforin, the cell lyses, and it can no longer infect us. These are real microscope pictures showing up here on the left. Here is a cancer cell that is infecting the body, and all these little white puffy cells are those cytotoxic T cells. And you can see as cancer cells are being attacked, they actually fight back. All of these little, looks like rays or little needles coming out. Those are trying to actually puncture. You can see one gets hit right here with one of those little spears from that cancer cell. So they're literally battling. And then the after picture here, it's flattened after the whole perforin lysing thing, and the white blood cells kind of conquered over this cancer cell. Pretty crazy. Okay, so T cells, again, some being cytotoxic, ones that were proliferated during that activation process. They might not necessarily respond in this first exposure. We have memory T cells that will not be involved in this first battle, but if we were exposed again to the same virus, then they would jump in super quick with a very, very strong immune response, and we couldn't then get a full infection from the same thing a second time. Okay, on to the B cell specifics then. We said the B cell is going to be most beneficial to us if it's activated by a helper T cell. But you see that usually B cells are going to encounter the foreign antigen somewhere else first. And so that's what's happening up here, is just a B cell wandering around the body, encounters the foreign antigen that its receptor shape is complementary to. And so here it's encountered the foreign antigen and it gets activated, but it's not a full-blown, strong, activated immune response until you can see it's kind of partially activated here. It's processed the foreign antigen. The most effective B cell will be then like with extra help from the helper T cell. And so here you can see this activated helper T cell that has the same or similar receptor for this same foreign antigen, encounters this partially activated B cell, and then move this out of the way. This B cell is fully activated. You can see that helper T cell releases cytokines, that kind of chemical signaling, chemical cell-to-cell communication again to reinforce this full-blown activation of a B cell. It's going to attract more macrophages and other leukocytes to the area in this inflamed area of infection. And as then the B cell proliferates, makes plasma cells, makes antibodies, the antibodies are doing their thing and helping to wipe out this pathogen. The macrophages that are in the area will then kind of digest those dead pathogens as this whole process occurs. Here we go in B cell activation. As the B cell encounters that antigen that fits its antigen receptor, here's that B cell talking to that helper T cell and like reinforcing the activation of the right B cell. And this is the like best pathway in order to get the strongest immune response. You'll hear a lot of talk about like how long does your immunity last for certain things, and oftentimes that has to do with how strong of an immune response you had to that pathogen. So again, preferably we're going to go from APC to helper T cell to B cell, and that B cell had hopefully already also encountered that same foreign antigen, and so you get the most full-blown response of the immune system when you take this pathway. Similar to picture to what we saw before, there's lots of different receptor shapes on our B cells. We have to make sure we get the appropriate B cell activated, the one that has the complementary receptor. That's the one that's activated. That's the one that divides by mitosis. Get that huge clone army. Again, like 80 to 90% of all the clone babies from the activated B cell become what we call plasma cells. They're making the antibodies. And again, those antibodies have a similar shape to the original receptor. Some of those babies, those clone cells, do not get involved in this first fight. Those are memory cells. So you do have memory B cells, memory T cells that don't engage in this initial infection. They just hang back in case later on in life there's another exposure. So some activated B cells, like we said, become plasma cells. Those plasma cells are what actually make the antibodies. It's not the B cell itself. So antibodies then can react against the antigen that stimulated their production in the first place. So we'll talk a little bit today about antibody actions too. And then we said there's also memory B cells. They stand by for future infection. Another graphic to go back and look at to see kind of that activation of the B cell, proliferation by mitosis, plasma cells make antibodies, those antibodies get into all the body fluids, and then there's really no chance of infection anymore. The antibodies inundate the body, inundate the pathogen. So getting to the end here with types of antibodies, the more technical term for antibody is immunoglobulin. So we abbreviate that with IG, and there's 5 major types. The way I remember them is GAMED, so there's G-A-M-E and D. The ones that I've been thinking about the most recently are G and M, because those ones are most involved when we get infected with a virus, and they are responsible for what we call a complement function, which I'll get to here in a second. But an antibody is just another protein with a very specific shape. They all are kind of Y-shaped, but the most important active binding site or antigen binding site is kind of up at the top prongs of the Y. So basically, an antibody has 2 sites where it can bind to the foreign antigen, and we see this occur, and we actually looked at it with blood typing. When you get the wrong blood type in a blood transfusion, the antibodies use kind of 2 prongs to kind of handcuff red blood cells together with the antigen that it can bind with. So if there was one cell with an antigen here and another cell with the antigen there, those 2 would now be hooked together, kind of like handcuffs, and when they're stuck together, they can't really function normally. You can see zoomed into this antigen binding site, how it would have a very specific receptor shape and a really tight fit there to make it effective. Here you can see immunoglobulins G, A, E, D, and M. You can see this M one is kind of doing this coating idea, where you've got like a toxin here, or like a protein produced by a virally infected cell, something that'd be causing a bunch of immune response. And you can see how the antibody has kind of grouped all around it and kind of coated it. I think of, and I usually in class would show the little video clip. You guys know The Incredibles. Can you imagine that scene where Mr. Incredible is like in that volcano and he's running down that bridge, and then those bullets are shooting at Mr. Incredible? Those bullets are kind of like antibodies, and if Mr. Incredible was like a pathogen, those bullets like are sticky bullets, and they hit him and then they swell up and he's like disabled. That's what I think of when I think of immune antibodies and how they work. How they disable a pathogen. So if we really break down the way that antibodies work, The first main way is by directly attacking the antigen, by like sticking to the outside, by handcuffing together pathogens with this antigen, or by a precipitate, actually pulling those pathogens out of solution because they're dissolved in all of our body's liquids, like the rest of our cells are. So if we're clumping together, kind of that handcuffing idea, we call that agglutination, like we talked about with blood typing. They can form insoluble masses and like precipitate. If you guys remember in chemistry class, when you take two liquids and you mix them together, the chemical reaction happens and you get kind of cloudy liquid as the product. And then you could like pull the liquid out and have like a powdery precipitate left. So you're basically pulling it out of a liquid. Antibodies can do that, or they can neutralize, like create a coating and neutralize, kind of like that Mr. Incredible example is another way that antibodies work. But the end result, as you've kind of decapacitated the pathogen with these antibody actions, it's still in your body. We still need to like get that garbage out. And so the ultimate result is phagocytosis. Again, macrophages, monocytes, neutrophils, those phagocytic cells have to go in and they're like the garbage disposals also. And anything that's like a dead-end pathogen now has to be eaten up and discarded. We got to get all the remnants out of our body. Here you can see that agglutination idea. The pathogen is here, maybe bacteria with this little yellow bump antigen. You can see the Y-shaped antibodies grabbing and linking them together. They clump, and again, macrophages are drawn to this, and they're going to then eat it up, dispose of it. Same idea here. Oh, whoops. Sorry about that. The second way would be kind of this two-step process of activating a complement, and IgG and IgM are usually the ones involved with this, and these are the ones we see most active in people with COVID-19 too. When you take that antibody test for COVID-19, you're looking for these two specifically. I think the IgG spikes first, and then the IgM comes along a little bit later, antibody-wise. But those ones make a complement. A complement is a group of proteins that work together to produce a variety of effects, but it ends up being very similar to what perforin does, that chemical release by those killer T cells. So with a complement, we end up having IgM and IgG get on the surface of the pathogen or virus-infected cell, for example. There's a reaction that occurs with that foreign antigen on the surface, and that creates this rupture, this pore, and as there's holes in the cell or pathogen, it floods with liquid and salts, it makes it all swollen and big, and then it explodes. And so the complement ends up basically being the same idea as like what perforin does action-wise, and then again, phagocytosis occurs and cleans up all the debris left over. Some pictures showing different actions of antibodies, this complement system shown here. But basically, in the end, it ends with this lysis. If you look at the bottom, showing the lysis where holes are perforating, and it would then swell and explode. Opsonization ends up being like coating the surface. It says surface antibody attachment. All these little red antibodies coat it on the outside, and it basically tags it and disables it so that macrophages can come along and engulf it. Makes it like easily engulfable, and then the macrophage basically uses its lysosome and digests it till it's dead. And macrophages, they do have special little receptors for endocytosis, that receptor-mediated endocytosis, where they've got their little structure on the outside where antibodies can actually connect, and it helps them find easily, this looks like a bacteria, to engulf and then destroy. So antibody actions, as your body creates a ton of antibodies, you're starting to feel better as soon as you have antibody production anyways. And as those antibodies flood your body, a lot of the symptoms go away. Maybe your fever starts to go down. But you're still going to have kind of the residual effects like mucus and things like that. We can see here that you've got white blood cells that are in the area trying to clean up a wound with mast cells. Mast cells are releasing histamine, which makes everything leaky, right? Lots more mucus in the area. Antibodies, they promote inflammation, all of those like heat and pain, redness, swelling, those types of things. And areas of infection are going to persist until we fully get rid of the infection and the antibodies start to subside. More pictures showing the different types of antibody actions. Let's see, this opsonization where we're like coating the pathogen itself. Here you can see this one with bacteria, antibodies all throughout, with anti-agglutination when we're like handcuffing them together. The complement system down here, but they all result in more phagocytosis. So ultimately, you're just getting more macrophage, monocyte, neutrophil action in the end to get rid of the pathogens once they're disabled by antibodies.